Good day, this is Professor Will, CBMD PhD. Today is Wednesday, March 25th, 2020. It is 3:50 and 31 seconds a.m. Eastern Standard Time. And this is a continuation of my many lectures on corona. So let's get started here. Coronavirus outbreak raises question. Why are bat viruses so deadly? Bats' fierce immune systems drive viruses to higher virulence, making them deadlier in humans. Date February 10, 2020. Source University of California, Berkeley. Summary. A study of cultured bat cells shows that their strong immune responses, constantly primed to respond to viruses, can drive viruses to greater virulence. Modeling bat immune systems on a computer. The researchers showed that when bat cells quickly release interferon upon infection, other cells quickly wall themselves off. This drives viruses to faster reproduction. The increased virulence and infectivity wreak havoc when these viruses infect animals with tamer immune systems, like humans. It is no coincidence that some of the worst viral diseases outbreaks in recent years, SARS, MERS, Ebola, Marburg, and likely the newly arrived 2019 nCoV virus originated in bats. A new University of California, Berkeley study finds that bats' fierce immune response to viruses could drive viruses to replicate faster, so that when they jump to mammals with average immune systems, such as humans, the viruses wreak deadly havoc. Some bats, including those known to be original source of human infections, have been shown to host immune systems that are perpetually primed to mount defense against viruses. Viral infection in these bats leads to a swift response that walls the virus out of cells. While this may protect the bats from getting infected with higher viral loads, it encourages these viruses to reproduce more quickly within a host before a defense can be mounted. This makes bats a unique reservoir of rapidly reproducing and highly transmissible viruses. While the bats can tolerate viruses like these, when these bat viruses then move into animals that lack a fast response immune system, the viruses quickly overwhelm their new host, leading to high fatality rates. Some bats are able to mount this robust antiviral response, and also balance it with an anti-inflammation response. Our immune system will generate widespread inflammation if attempting this same antiviral strategy, but bats appear uniquely suited to avoiding the threat of immunopathology. The researchers note that disrupting bat habitat appears to stress the animals and makes them shed even more virus in their saliva, urine, and feces that can infect other animals. Heightened environmental threats to bats may add to the threat of zoonosis. The research project explores link between loss of bat habitat and the spillover of bat viruses into other animals and humans. The bottom line is that bats are potentially special when it comes to hosting viruses. It is not random that a lot of these viruses are coming from bats. Bats are not even that closely related to us, so we would not expect them to host many human viruses. But this work demonstrates how bat immune systems could drive the virulence that overcomes this. Vigorous flight leads to longer lifespan and perhaps viral tolerance. As the only flying mammal, bats elevate their metabolic rates in flight to a level that doubles that achieved by similarly sized rodents when running. Generally, vigorous physical activity and high metabolic rates lead to higher tissue damage due to an accumulation of reactive molecules, primarily free radicals. But to enable flight, bats seem to have developed physiological mechanisms to efficiently mop up these destructive molecules. This has the side benefit of efficiently mopping up damaging molecules produced by inflammation of any cause, which may explain bats' uniquely long lifespans. Smaller animals with faster heart rates and metabolism typically have shorter lifespans than larger animals with slower heartbeats and slower metabolism, presumably because high metabolism leads to more destructive free radicals. But bats are unique in having far longer lifespans than other mammals of the same size. Some bats can live 40 years, whereas a rodent of the same size may live 2 years. This rapid tamping down of inflammation may also have another perk: tamping down inflammation related to antiviral or immune response. One key trigger of many bats' immune systems is the hair trigger release of a signaling molecule called interferon alpha, which tells other cells to man the battle stations before a virus invades. Researchers were curious how bats' rapid immune response affects the evolution of the viruses they host. So they conducted experiments on cultured cells from 2 bats and as a control 1 monkey. One bat, the Egyptian fruit bat, Rosetus aegypticus, a natural host of Marburg virus, requires a direct viral attack before transcribing its interferon alpha gene to flood the body with interferon. This technique is slightly slower than that of the Australian black flying fox, Pteropus electro, a reservoir of Hendra virus, which is primed to fight virus infections with interferon alpha RNA that is transcribed and ready to turn into protein. The African green monkey, a Vero, cell line does not produce interferon at all. When challenged by viruses mimicking Ebola and Marburg, the different responses of these cell lines were striking. While the green monkey cell line was rapidly overwhelmed and killed by the viruses, a subset of the Rosetta bat cells successfully walled themselves off from viral infection, thanks to the interferon early warning. In the Australian black flying fox cells, the immune response was even more successful, with the viral infection slowed substantially over that in the Rosetta cell line. In addition, these bat interferon responses seem to allow the infections to last longer. Think of viruses on a cell monolayer like a fire burning through a forest. Some of the community's cells have emergency blankets, and the fire washes through without harming them. But at the end of the day, you still have smoldering coals in the system. There are still some viral cells. The surviving communities of cells can reproduce, providing new targets for the virus and setting up a smoldering infection that persists across the bat's lifespan. This suggests that having a really robust interferon system would help these viruses persist within the host. When you have a higher immune response, you get these cells that are protected from infection, so the virus can actually ramp up its replication rate without causing damage to its host. But when it spills over into something like a human, we don't have these same sorts of antiviral mechanisms, and we could experience a lot of pathology. Researchers noted that many of the bat viruses jump to humans through an animal intermediary. SARS got to humans through the Asian palm civet, MERS via camels, Ebola via gorillas, and chimpanzees Nipah via pigs, Hendra via horses, and Marburg through African green monkeys. Nonetheless, these viruses still remain extremely virulent and deadly upon making the final jump into humans. It is really important to understand the trajectory of an infection in order to be able to predict emergence and spread and transmission. Good day, this is Professor Will, CBMD PhD, on infectious disease and pathophysiology. Well, I hope you enjoyed listening and have a great day.